Yes, students. Now we'll continue with the next salient feature of HGP, that is the seventh one. Repeated sequence make up very large portion of human genome. Yes, I told you in generation of physical and genetic maps. That is, the map is DNA sequences are joined in two ways. One is restriction endonuclease enzyme recognition site. Other one is microsatellite. That is, sequences of DNA in human gene are repeated at frequent intervals. So that what can say sequences in gene are repeated. That's what we say. That is, repeated sequences make up very large portion of human genome. At many intervals, the same nitrogenous bases are repeated. Very simple to tell you. You will be studying about what is that mRNA. mRNA has got poly A tail. It is made up of repeated number of adenine nitrogenous bases. That is, around 200 adenine bases have been joined continuously. This is one example for repeated sequences. Like that, even the DNA has got nitrogenous bases which are repeated at many regions. So therefore, in human gene, these sequences are very more. The next characteristic feature is repeated sequences are stretches of DNA that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct functions, but they shed light on chromosome structure, dynamics, and evolution. Yes, very important is this. Yes, I was telling you earlier part of this topic, that is less than 2% of genome codes for protein, remaining 98% they are present. Why? Might be these are the region, that is they are repeated sequences of DNA where the sequences are repeated 100 to 1000 times. So now, what is the role of this? They do not code for any protein. They don't have any functions at all as far as synthesizing protein is concerned. Then why they are still present? That's a very big question. So therefore, these are present. These are present. Their presence is required to understand the structure of chromosome. That's a very important to understand functioning of chromosome, because you know that human contains 30,000 genes. All genes will not function simultaneously, or they are not going to function together. Some genes may function at one time. Some genes, as you'll be studying in lac operon, switch on and switch off will be there. So how this switch on and switch off is going to take place? So that can be understood by this, what can say, existence of this repeated sequences. And likewise, this is very, very important. That is, this is going to speak about human evolution. So that's a very important role of this HGP. Then chromosome 1 has most number of genes, that is 2968, and Y has fewest genes, that is 231 genes. Yeah, this is also very, very important. We have studied that human being has got 23 pairs of chromosomes or 46 chromosomes. Hope so. You remember I have shown the chromosome pictures while explaining you chromosomal anomalies. And if you see there, all chromosomes are of not same length. Why? It depends on the number of genes that chromosomes are made up of. So here they say that chromosome 1 is the biggest, might be because it has got 2968 genes. So most genes are present in chromosome 1. Whereas chromosome Y is the smallest one because it has got less number of genes. So therefore, by seeing the arrangement of genes, we are going to know the shape and size of chromosome also. Then the next and the last ultimate salient feature of HGP is, yes, scientists have identified about 1.4 million locations where single base DNA differences are known to occur, where the single base DNA differences are called as single nucleotide polymorphism, which is pronounced as SNPs. So this SNPs existence is important because it speaks about the process of finding chromosomal locations for disease associated sequences and tracing human history. Very, very important part is this. That is, in human gene, there are more than 1.4 million locations where just one nitrogenous base will differ when you compare one person with another person. So this what can say locations are called as SNPs. So they are very, very important to know about diseases. Yes, you have studied about chromosomal diseases, you have studied about Mendelian disorder, why they are caused. For example, you take sickle cell anemia, you take phenylketonuria. In all those cases, the disease has occurred mainly because of mutation of one nitrogenous base pair. So you take sickle cell anemia. So glutamic acid has been replaced, sorry, valine has been replaced by glutamic acid because of just mutation of one nitrogenous base pair. So therefore, this what can say SNPs are very, very important to find out whether human being is normal or abnormal, whether he has got any inborn errors of metabolism, whether he has got any chromosomal anomalies and so on. To trace about that, this single nucleotide polymorphic regions are very, very important. Not only that, they are going to trace about human history. Yes, we say all human beings belong to same species, that is Homo sapiens. But still, you might have observed there are different races in human being. That is, for example, you take Negro, you take albino, then Mongolian, Indian, Chinese. There is variation in the characteristic feature of this people. Negroes are extremely black, albinos are extremely white, Indians are brown, then Mongolians between brown and white, and Chinese. Their physical appearance is somewhat different. Even character is somewhat different. Even if you compare IQ, it differs from one race to another. So how these changes have occurred? So how to know about that? That can be understood by knowing this single nucleotide polymorphic sites on human genome. So therefore, these are the 10 salient features of human genome project. Like this, there are many, but for your level, this 10 what we can call salient features are more than enough. Dear students, remember this is a very, very important part. You have got one 5 marks question. List out the salient features of HGP. So naturally here you have got problem with goals and salient features. Don't get confused. Goals, they were set up before making human genome project. Salient features, they have been identified after making human genome map. So therefore, if question is list the salient features, it's understood that you have to write all the 10. But if the question comes list any 5 salient features, you have got choice, you can write any 5. Okay, so this is with regarding to salient features of HGP. In the next slide, we will be speaking about applications of HGP. Until then, goodbye.